All right, welcome back, everyone. It's 1:15, so let's go ahead and begin our afternoon session. So the part 2 session is entitled Beyond Screening, Zebrafish as a Model for Developmental Mechanisms at the Cellular and Molecular Levels. And our afternoon speakers are Dr. Jennifer Panlilio, Woods Hole Center for Oceans and Human Health, and Dr. Dan Wagner, from the University of California, from the University of California, San Francisco. After their presentations, we'll have 30 minutes for committee discussions like we did this morning, and then we'll take a short break and then move on to the last part of the session, a discussion with the committee and all 4 of our invited speakers. So I'd like to now introduce Dr. Jennifer Panlilio from Woods Hole. Her research interests include circuit neuroscience and neurotoxicology. Recent publications based on her graduate work feature the use of zebrafish larvae to study the effects of an algae-produced neurotoxin domoic acid on outcomes of neurobehavior and neuronal development. And her title of her talk today will be Use of a Zebrafish Model to Investigate How Low Doses of Domoic Acid Affect the Developing Nervous System, including windows of susceptibility, structural and molecular changes in nervous system tissues, and links to behavioral alterations. Dr. Panlilio, welcome, and thank you for speaking today.
Wonderful. Thank you so much for that introduction. So let me share my screen. Please let me know if you don't see the slides shortly.
Okay. Yes, we see, or not, now we see presenter view. Perfect.
Wonderful. Okay, so it's a pleasure to speak to you all about my research on essentially how we can use zebrafish to identify the developmental mechanisms of neurotoxicity. So for this talk, I'll focus primarily on uncovering the mechanisms of neurotoxicity that occur from exposure, particularly to a harmful algal bloom toxin known as domoic acid. But firstly, what is harmful algal blooms and what's a harmful algal bloom toxin? So it's probably familiar in the West Coast, but harmful algal blooms are these mass accumulations of algae that are defined really by their adverse societal impacts rather than by any strict scientific definition. So there are plenty of ways that harmful algal blooms, or HABs, can cause harm. And so some of these HABs produce toxins that directly affect human health. And so one particular harmful algal bloom toxin that I study is called domoic acid. So this toxin is produced by a diatom. So this is a type of phytoplankton that you see over here. And then shellfish and other seafood we eat can then accumulate domoic acid as they're filter feeding for these phytoplankton, which in turn we then can consume. And so when domoic acid is consumed in high enough doses, particularly in adults, it can cause what's known as amnesic shellfish poisoning. So symptoms from amnesic shellfish poisoning include gastrointestinal issues, memory loss, coma, and even death in the most severe cases. And so really, to prevent acute toxicity in adults, we do have a regulatory limit set at 20 milligrams of domoic acid per kilogram shellfish tissue. But I do want to emphasize here that this limit is based on protecting adult humans from acute toxicity and those severe overt symptoms that I just previously mentioned. However, we do know that humans are regularly exposed to domoic acid below these limits, and that furthermore, humans can be exposed to these toxins during potentially more susceptible periods of early development. And so what do we know about domoic acid? So we know there's now ample evidence that it is a developmental neurotoxin. So we know, for example, from rodents, that both prenatal and postnatal exposures can lead to long-term behavioral defects, aberrant neural activity, and pronounced changes in brain tissue architecture. It's also been notably shown to heighten sensitivity to other toxicants that the rodents are exposed to later in life. We've also have used zebrafish in the past, and we show that we have previous work has shown that zebrafish exposed to high doses of domoic acid very early in development. Here, it's the 1K cell stage, causes behavioral phenotypes that are consistent with other animal models that are acutely exposed to high doses of domoic acid. However, no zebrafish studies have been done up until, we published ours on more lower level doses that may be potentially more relevant for human exposures. And further, there was no previous studies that looked essentially into those more potential windows of susceptibility for exposures. So knowing this, our studies really wanted to address these knowledge gaps. And so there were these three primary objectives. And so the first is to identify specific windows or developmental windows of susceptibility to domoic acid exposure, to identify the functional consequences for behavior, and then to determine whether there are specific cell types that are targeted by exposures to domoic acid. And so to accomplish this, and this is what this meeting is about, right? We utilize the strengths of the zebrafish model. So one thing that's really nice is zebrafish have these really simple and quantifiable behaviors. And so we've chosen one behavior in particular where the neural circuits that underlie the specific behavior is very well characterized. And so what that allows us to do is it allows us to link that potential behavioral deficit to the underlying neural circuits that drive it. And finally, we've also heard this, but they have transparent larvae that develop externally. So what this really allows us to do is it allows us to directly image cellular processes that may be perturbed over the course of early development. And we can do this in real time as the animals are living. So to identify the critical windows of susceptibility, we exposed fish in discrete periods in early development through a different method of exposure than what we've heard about, but we've used intravenous microinjection. And then we to identify the mechanisms of toxicity, we then took this multi-level approach where we measured the behavior as the readout of toxicity, and then assess the potential structural changes in the neural circuit that underlies this behavior. And then finally, we link these to specific cellular targets and molecular effects from exposures. So in particular, I exposed zebrafish to domoic acid, again, during these discrete periods in early development. And the way we could do this is through intravenous microinjections. So the microinjections were done into their caudal vein. So essentially, we took the little needle and we could find the caudal vein and inject them over time. And we did this at 1, 2, and 4 days post-fertilization, or DPF. And so we decided to do this over the more common route of exposing them, which is through chemicals in the water, that immersion type exposure that Dr. Padilla spoke about, because we wanted to ensure domoic acid was entering in the fish. And it also really allowed us to mimic that single acute exposure during these very discrete periods of early development. And so just to back up a little bit, why did we choose these 3 periods? I targeted these 3 key early stages because they mark 3 neurodevelopmental stages that are quite important for the fish. So for example, exposures at 1 day post-fertilization is when neurons and glia cell precursor cells are first specified. It's when majority of those early sensory and motor neurons differentiate. And then at 2 DPF, this is the time where that would target a specific glial cell called the oligodendrocyte, which I'll talk about quite a bit. This is when it starts to migrate and differentiate and starts to wrap axons across the central nervous system. And then finally, exposures at 4 days post-fertilization correspond to those later developmental periods where most of the cell types in that early nervous system are already specified. So following these exposures, I then did a detailed assessment of the startle response behavior in the larval stages between 5 to 7 DPF. And during the same larval period, I also imaged the different cells and structures that make up the circuit that drive this response. And so I mentioned startle, so that's the behavior we're looking at. So what is it and why are we using it? And so to assess the functional effects of domoic acid, so we're using the startle response behavior. And so the larval startle response behavior occurs in response to a sudden and intense stimulus. It's quantifiable and it really requires proper sensory processing and motor control. So I'll argue it serves as a tool to determine whether these processes are disrupted by exposure to domoic acid. Finally, it's driven by underlying neural circuits that are pretty well known. So it makes it a really powerful behavior to study because, again, it can allow us to link these behavioral results to the anatomy and the cell types that are involved in the circuit. And so to assess startle behavior, essentially these fish are placed in these 16-well plates that sit above a speaker, and their startle responses are recording using a high-speed camera above. Okay, so here's just a zoom in on a single well that you see over here, and the larvae is essentially divided into 3 segments as encoded by these 3 different colors. So the changes in the curvature as the fish undergoes startle are estimated by the changes in angle between these 3 segments. So what I'll do is I'll play a video, so you'll see the video over here. And what you see to the right of the video are the estimated changes in curvature as the fish undergoes a startle response. So note that there's this initial really large bend angle. This is that C bend over here, followed by a corrective angle. Then the fish starts swimming away and then eventually stops. So I'll play that just one more time so you can see that. So that first really large bend angle and the corrective one. And so what's nice about this is from these videos, we can get some really interesting kinematic attributes. So for example, we can get latency, right, which is the reaction time from when the stimulus is produced to when the fish reacts. We can get, for example, that maximal bend angle from that first bend. We could get like the angular velocity, et cetera, et cetera. So we use this behavior, right? And so we expose zebrafish at different developmental time points, and then we assess their startle behavior kinematics. And so what we found is that fish exposed to domoic acid, particularly at 2 dpf, from the lowest dose all the way to the highest dose, had reduced bend angles relative to controls. In comparison, the fish exposed to domoic acid earlier at 1 dpf or later at 4 only had reduced bend angles at the higher doses, and the bend angle reduction was a lot less severe compared to those exposed at 2 dpf. This is also true for other kinematic attributes we looked at, such as the maximum angular velocity that led to that bend as well. So we know that there's this behavioral deficit that results from domoic acid exposure, and that it is most true from when we're exposing fish, particularly at this 2 dpf time point. Again, just to emphasize, I love this behavior because we know a lot about the underlying cells and their connections that drive it. And so knowing that there's deficits in startle, we then sought to look at the anatomy of the circuit. And one thing we started looking at first were myelin sheets. And so startle responses are extremely fast. This is why we really need those high-speed video cameras to actually record them. So they happen between 50 and 50 milliseconds after a stimulus is given, which also requires extremely fast conduction velocities through the axons as essentially the signal propagates down the axons and innervates the muscles across the trunk. And so to allow for this rapid propagation of the signal, axons within the startle circuit are heavily myelinated. And in fact, this neuron that I'll talk about a little later is the most heavily myelinated neuron in the nervous system of the fish. And so we assess myelination in the spinal cord, again, because proper myelination is required for proper startle. And so to accomplish this, we again employ the power of the zebrafish model, right? So we have this transgenic fish that have myelin sheets that are labeled by GFP. And because zebrafish are transparent when they're younger, we can again image these structures in living animals, right? And so here's just a fish on its side. Here's just a cartoon of a cross section of its spinal cord. And in control fish, what you see is that there's 2 regions here. This is the ventral region of the spinal cord, and this is the dorsal. And what you see is it contains myelin that is both abundant and elongated. Now, again, so that's our control fish, right? And using these transgenic fish, we can score myelin phenotypes visually from those, again, that look like this beautiful control, as you see up here, to those that essentially have these disorganized sheets where essentially the myelin and the axon doesn't follow the track perfectly, all the way to the most severe form where there are very, very sparse sheets, and they contain all these like very unusual circular features, which I'll get back to later about what we think those are. Okay, so what we did is we, again, exposed fish to domoic acid. And what I'm showing you right here is an exposure to an intermediate dose. And then we did this over different developmental time periods. And what we see is that the highest proportion of fish with myelin defects occur when domoic acid is exposed at 2 DPF and 2.5 DPF. And those exposed at 2 DPF have a larger proportion of the more severe myelin defects. So these levels right here. So we also tested a range of doses and during a range of different developmental times, and we found that even down to 0.09 nanograms, which is the lowest dose we tested, we still see an effect at 2 GPF, while not seeing this for fish exposed at the 2 other time periods you see up here. So note, though, that there are gaps in the developmental times we were, and for example, there are places we were unable to test, like this one. So for example, within our highest dose of 0.18 nanograms, we did not test fish exposed at 4 DPF because they did have other phenotypes that made them untestable. And so I do want to bring up that point, and I think it's also important because one such phenotype, severe phenotype these animals have, but is essentially widespread brain necroses, where you see essentially their head, because they're transparent, it looks like a cloudy brain, if you will. And so in this case, right, over 40% of the embryos injected specifically at these later periods, so at 4 DPF, show this very severe phenotype. So it seems that at higher doses, those overt severe phenotypes can occur at the later stages of exposure. So both the behavioral data and the myelin sheath data both now point to 2 DPF being an important period for domoic acid exposure, especially exposures at the lower ends of the dose range. And so we wanted to continue to investigate whether exposure at 2 DPF disrupts other important players in the neural circuit. So to do that, I'll step you through the different players that make up the startle circuit that we know pretty well. So in addition to myelin sheaths that are required for that rapid execution of that startle response, there are other things that are involved, right? So auditory and vibrational stimuli are first detected by the hair cells that occur both within the inner ear and within the neuromasts that make up the lateral line. And so the mechanical deflection of these hair cells then lead to the activation of sensory neurons. And then these sensory neurons can then send information to the hindbrain. And so in the hindbrain, cells like the Wagner cell, along with other homologs, integrate all the sensory information. And once the Wagner cell reaches threshold, it then fires a single action potential, which propagates down its axon that extends into the spinal cord. And then what this results in is that as the action potential rapidly travels down the spinal cord, it activates primary motor neurons along the way. And because the signal propagates so quickly, this leads to these really fast unilateral muscle contractions and these deep bend angles that you saw in the video previously. So, okay, so let's step through the circuit. Let's look first at the sensory side. So let's look at the neuromasts and the inner ear hair cells as in addition to the other sensory structures. Okay, so to assess neuromasts, we used a live cell staining dye called DASPI. And so essentially, this is one of those cases where you can put a zebrafish in a dish and expose them in the water to this live staining dye. They will take it up. And what we found is that domoic acid exposed fish have the same number of neuromasts, both in their heads. So we looked at the number of neuromasts in the head, as well as their trunk region over here. So it doesn't seem, at least in terms of absolute neuromass count, that domoic acid alters that. We also looked at various sensory neurons, their axonal tracts, and found no differences. For example, here's an image of a zebrafish. These are fixed fish, so these are now antibody staining, at least on the top. And what you'll see here is that we see no differences. The inner ear is outlined in this like teal dash outline. We saw no differences in the inner ear hair cells. We also, using a live imaging of another transgenic, also found no noticeable differences in both the lateral line, the neuromasts again over here, these are the neuromasts, as well as that lateral line, which is the ganglia or that nerve, the nerve connecting all these neuromasts over here. So I like to point this out, that at least at this stage and at this resolution, it doesn't seem like domoic acid is targeting the sensory system directly. So again, so that's the sensory system. And so after sensory information is collected, right, by the inner ear hair cells and the lateral line, it then sends it to the mouth nerve cells and its homologs. So we then assessed for the presence of this specific mouth nerve cell, which is this teal one in this cartoon over here. And so here we use antibody staining to show that while all the controls we imaged had these two mouth nerve cell pairs, so here is one pair going this way, the axon crosses the midline and goes down contralaterally. So it has these two beautiful mouth nerve cells here. Most domoic acid exposed fish had no mouth nerve cells. So here is an image where you just don't see the mouth nerve cells here, but there were a few with two, but majority of them did not have mouth nerve cells. The other thing we can look at in the same tissue, right, is we can count the hindbrain tracts. So that would be these lines that you see over here. And when looking at these tracts, we found there's no significant difference between the domoic acid exposed fish and the control. So it, again, emphasizing that it isn't all neurons that are altered by domoic acid, but potentially very specific targets. Okay, so these findings indicate that exposure to domoic acid led to these measurable startle deficits, particularly exposures at 2 dpf. And in conjunction with these, we also see that there's a loss of this mouth nerve cell that's really important for startle, as well as myelin defects during the larval period at 5 dpf. So note that all of these endpoints were taken during the larval stages, which is after nascent myelination has occurred, which occurs between it starts at like 2.5 and ends mostly at 5 dpf. So it's after this nascent myelination has occurred, and also it's well after exposures at 2 dpf, right? And so we then wanted to look more at the initial cell targets and the initiating events that perhaps may contribute to all those later larval phenotypes we were seeing. So recall, right? So in the larval stages, we both saw the loss of that mouth nerve cell along with its axons in the spinal cord, as well as myelin defects. So we know, of course, that myelin cannot, so here's like my little cartoon, right, of an oligodendrocyte, which wraps myelin around axons. So we know, of course, that myelin cannot wrap around axons that are just not present. So if an axon, for example, is lost first, we expect there to just be not as much myelin around, right? And there might be myelin defects. But we also know that myelin provides really important metabolic support for axons. And so the loss of the myelin could then also lead to axonal defects. So the question then becomes, what happens first, right? Do we lose myelin first and then the axons follow, or vice versa? And so to address this question, we first wanted to see how domoic acid exposure affects those initial stages of myelination. And again, I'll emphasize here like that I think the power of this model is we can not only easily image fish because they're transparent and they're small, but we can also visualize cellular processes that are occurring in real time. So that's what we did here. We're essentially performed time-lapse imaging with transgenic fish. So these are live fish. Where we looked for and we observed myelination actually happening within their spinal cords. And so what you're looking at here is the cartoon on the left is essentially, this is a transgenic fish where our cell bodies of the oligodendrocytes, those are the cells that make myelin, are labeled in red, and their membranes that become myelin are labeled in green. And so what you'll see here, this is a time-lapse video that occurs over the course of around 12 hours. And so on the top, you'll see the controls are forming these beautiful myelin sheets. So there's those little thick processes, elongated processes here and here, for example. In comparison, domoic acid exposed fish primarily do not. So what you'll see here is, so here they are, right? These oligodendrocytes have these red cell bodies, and instead of traveling around and starting to myelinate, they tend to form these shorter sheets, right? So you don't really see those elongated sheets that you see here. And they also form these really strange, I'll play that one more time. Oops. They tend to form these really strange circular features, which again, I am pointing out because this becomes relevant later to what we think these phenotypes are. All right, so now we know the initial stages of myelination is perturbed. And so now we turn more specifically to looking at, again, those oligodendrocytes, which is that lineage of cells that make myelin. So we looked at how domoic acid perturbed oligodendrocyte development a little bit after the time myelination commences. So we used another fish line. So we have a lot of fish lines through this study. And this fish line essentially labels oligodendrocytes that you see here. And so what we did is we quantified the number of myelinating oligodendrocytes in the spinal cord during the period of myelination. So as I said, so each of these individual points is an individual oligodendrocyte that we can quantify. And then on the right, you'll see each point in this graph represents the number of oligodendrocyte counted from an individual fish that was exposed either to no domoic acid, or that was the control, all the way to the 2 doses of domoic acid you see here. And what you'll see is that there's a significant reduction in the number of oligodendrocytes, which is true for our medium range dose, but is particularly true for our highest dose of domoic acid. Okay, so now I'm building this model, right? So we know that domoic acid disrupts that initial myelination and leads to the loss of oligodendrocytes at 4-DPF. And the loss of the oligodendrocytes could conceivably contribute to the myelin defects we're seeing, because if there's a reduced supply, if there's less oligodendrocytes that can myelinate, this may just lead to the less myelin overall being formed, right? The loss of the oligodendrocytes could be because domoic acid is directly binding to and affecting these oligodendrocytes, because they do have the receptors to which domoic acid binds to. Okay, so now we've looked at the oligodendrocytes, right? But we also know that they have this Wagner cell loss. So let's revisit that. So we know that this occurs at the larval stages, but we wanted to determine whether these Wagner cells were absent shortly after exposure, but before myelination even commences, right? So all we had to do really was image domoic acid exposed fish and look for Wagner cells much earlier. And so using the same antibody, it's a neurofilament antibody, we're able to image the Wagner cell prior to myelination. So here's just a control brain to orient you. And here are, in these teal arrows, are those 2 beautiful large Wagner cells. And so while control fish always have 2 Wagner cells, domoic acid exposed fish have a range of phenotypes, but again, majority of domoic acid exposed fish don't have those 2 Wagner cells, even prior to when myelination commences. So now we know again that the Wagner cell loss occurs even earlier than myelination. Thus, the domoic acid exposed fish may be targeting, domoic acid, may be targeting these neurons first, and then altering the cellular environment in the spinal cord and in the brain prior to myelination, which may, have downstream effects from the oligodendrocytes and myelin later. So speaking of the cell environment, let's look at myelination in the spinal cord. So in a control animal, there's really this correct balance between the number of oligodendrocytes, as you see here. So this again is the cell that is responsible for myelinating in the brain and spinal cord. And so there is a balance between oligodendrocytes and the amount of axons they have to myelinate. But what happens when these axon targets are reduced? What if there's just less axons around? So the Lyons lab addressed this question by essentially using a genetic model that reduced axons within the spinal cord. And they found that the reduction of axonal surface area also led to the reduction in total number of oligodendrocytes. And they propose it's potentially because there's a feedback process which attempts to now correct the mismatch between the oligodendrocytes to the axons. What they also found is they found these appearance of these circular profiles. Remember that I've been pointing that out and saying, what are those? It's very strange. And what they found that to be this abnormal phenotype where they thought that it's due, and they actually show data to show that essentially instead of wrapping just axons, because there were not enough axons, these oligodendrocytes were starting to wrap neuronal cell bodies instead. So not just the axons, but the cell interface itself. And so they attributed, again, these effects to the mismatch within the axonal surface area to the number of oligodendrocytes present, because as there's less axons in the environment to myelinate, oligodendrocytes start to myelinate other things, right? Including, again, those neuronal cell bodies. So it is possible, right, that such a thing is occurring with domoic acid exposed fish too, and the loss of this mouth nerve cell, as well as other like large axons within the spinal cord, could contribute to the effects we're seeing with oligodendrocytes and myelin. So we wanted to see whether our circular features were very similar to theirs. Are these also incorrectly wrapped neuronal cell bodies? And so to identify what these circular features are, we used another transgenic fish. So here in magenta, this labels myelin, which is the oligodendrocyte membranes, and the green labels the membranes of neurons. And so and what you'll see here, right, this is a domoic acid exposed fish. There are these, again, these really unusual circular profiles that are produced by oligodendrocytes, primarily only in domoic acid exposed fish. And in green, in this picture over here, you're seeing these honeycomb-like structures that represent those densely packed neuronal cell bodies within the spinal cord. And in our merged image on the rightmost, you'll see that these circular oligodendrocyte membranes appear to be in the same location as the neuronal cell bodies. So suggesting that the oligodendrocyte membrane is wrapping neuronal cell bodies. And so this is just a schematic of just that, right? So there are oligodendrocytes that wrap, axons as they should, but in addition to that, they're now wrapping these cell bodies because there's just not enough axons around, presumably. So we then did electron microscopy, which will give us a little bit better resolution. And so to orient you, here's just a cross-section of a fish over here and here. So here's its whole trunk, and over here is its spinal cord. And if we zoom in on that, these circular, all these circles over these densely packed circles are neuronal cell bodies. And in control fish, right, most of these are just wrapped by a double membrane layer, which is what you would expect. And so that's true for a lot of domoic acid exposed fish. But in addition to that, we also have cells, neuronal cells, where there's more than just that double membrane, but there's a second outline around them, like here and here. So we think that these are also myelin inappropriately wrapping these cell bodies. And so with all this, we have a working model for how developmental exposures to domoic acid mediates toxicity. So the loss of the mouth nerve cell precedes disruptions in myelin. And then the loss of the mouth nerve cell contributes to this reduced axonal surface area in the spinal cord. And the loss of oligodendrocytes may result from that reduced axonal surface area as that feedback process attempts to correct the discrepancies between the axon surface area and the oligodendrocyte present. Myelin defects may also result in a reduced axonal surface area as oligodendrocytes start to myelinate neuronal cell bodies in the absence of just enough axons. So both these phenotypes have been found in the genetic model that lacks axons. So it's possible that all of these oligodendrocyte effects are secondary effects to the initial loss of the axons. I previously proposed that domoic acid may bind directly to the oligodendrocytes as well. And it is still possible that these cells do have receptors to respond to domoic acid. So it might be that these processes occur in concert. So with all of these elements can contribute to that observed aberrant startle behavior we see. And as you see, this model has a lot of question marks. So the receptors to which domoic acid binds to are expressed by multiple cell types in the nervous system. So again, conceivably, there could be multiple targets happening at the same time. So stepping back from all these very specific details, I want to discuss a little bit about what this means, that this could mean for human health. So this current research identifies a potential mechanism of domoic acid toxicity that occurs during a susceptible window of exposure in early development. So I found pronounced changes in spinal cord structure following domoic acid treatment. And so this highlights how domoic acid targets not just the brain, which is where a lot of people look, but also potentially the spinal cord. I also found that there are specific cells that were lost during domoic acid exposures. So humans don't have Wagner cells, but they do have a group of functionally equivalent cells. And so even if this research does not identify specific neurons that may be targeted in humans, I think it can provide us with clues as to what characteristics some cell types may have that may make them more susceptible to domoic acid toxicity. So perhaps neurons with really large axons that are excitable, that sort of span the range and have into the spinal cord, may be more sensitive to domoic acid exposure. So I also found that the loss of axons right before the big wave of myelination leads to really aberrant myelin phenotypes. And we could, of course, speculate whether domoic acid exposures in humans prior to myelination could also have important consequences. But what's interesting, right, is if myelination is truly a target of human exposure, it could have really important health consequences and exposure considerations, because unlike fish, myelination in humans spans from late prenatal development all the way into early childhood or even later, and thus leading to potentially a prolonged window susceptibility to domoic acid exposure. So of course this would all have to be tested, but it's just a framework for us to understand about exposures and how we think about it in fish, and we relate it to humans. And so just my last slide to step back from the domoic acid story, to just remind us that this could many ways be a model for how we assess really any neurotoxicant, right? We can leverage the zebrafish model to really do these more multi-level approaches where we start with a behavior, we step through the circuit, and we look at the cellular and molecular endpoints that underlie it. And so with that, thank you for letting me share this story, and I'm more than happy to take questions from everyone.
Well, thank you, Dr. Panlilio, for that wonderful presentation. We have just a few minutes now for clarifying questions from the committee, and then remember, we'll have more time for discussion later. So I see some raised hands already. Dr. Pessa?
That's a wonderful presentation. I really like the sort of methodological approach. You mentioned excitability, and did you actually measure the balance of excitation inhibition? Was that different at two days post-fertilization? Is it driving the axonal loss, I guess, is hyperexcitation?
Right. And, you know, I sort of glossed over the what we know about domoic acid. So domoic acid binds to a ionotropic glutamate receptor, so it is an excitotoxin. I did not measure that directly, but I would be very surprised if there were not changes in that for sure. And in terms of like why I speculate the Wagner cell, particularly at the 2-day post-fertilization time point, is susceptible, is there are papers that show that it actually will switch subunits right before 2-DPF for 2 subunits that are more excitable and that respond more. And it's a very specific subunit of a ionotropic glutamate receptor. So short answer is no, I didn't measure it directly, but I would be very surprised if it were not changed.
Thank you.
Yep.
Dr. Hertz-Picciotto, I see you have your hand raised too. You need to unmute.
So sorry.
Now we can hear you.
Yeah, my video is also a little lagged there. Yeah, I love this talk. You really explained very well your step-by-step approach. And I'm just, you know, I'm really struck about the myelination because, you know, at least 2 years postnatally, it's really immature, not complete. And what I don't know, and I don't know if you do, one question I have is, the brain versus the spinal cord in humans. Is the myelination happening more or less in both at the same time? Is that extended period also for the spinal cord? I think the 2-year that I had heard or that you constantly see in the literature is based on brain specifically. And then a second question I had also was, I'm interested in, you are interested in those lower doses that human beings get. And I'm wondering about the comparability of your exposure to the zebrafish, and how that might translate in terms of doses that humans are getting generally through seafood consumption, I presume.
Right. Wonderful question. So I will say you are right for the human stuff. My understanding of the ontogeny for myelination occurs from the brain. I do believe there is a paper that goes through different brain regions. I'm wondering if spinal cord's in there as well. So the short answer is I'm not sure for humans. However, for fish, nascent myelination occurs across both the brain and the spinal cord roughly during that embryonic period between 2 to 5 DPF. So it's a pretty relatively short window, but I guess if you stretch zebrafish out, it wouldn't be that short. But yeah, so great question about whether that's true for humans, and I'm not 100% sure. For the dose comparability, so I've thought about this a lot. And so the short answer is there's no direct comparison, partially because there's currently no information that I know of domoic acid concentrations in both human fetal tissues or even maternal. We just don't know what domoic acid is in maternal, human blood, the concentrations in serum. And so what we do know, for example, so all we really know is, unfortunately, the reason it was brought up to the forefront is that we know about an acute doses that can cause acute exposure in humans because of an incidental exposure that occurred in 1987, but that was acute and that was for adults. Our doses are comparable to if we assume that the embryos are 1.5 milligrams, which they can change, right? Our doses are comparable to doses used when they injected rodents subcutaneously postnatally to look at domoic acid, but they are higher than I would assume. We talk about route of exposure. They're much likely higher than what potentially a human would see, just because we're injecting it directly into the system, right? Depending on how if you were to, for example, eat domoic acid and it's an oral exposure, we find that oral bioavailability is pretty low. So if you were a young child, for example, you can think, for example, but they've also found, for example, that the concentration in fetal tissue is less than concentration in maternal plasma in rats. So all of this is to say, we're using zebrafish as a mechanistic model, realizing that there are some limitations in not being able to translate that dose directly.
Yeah, thank you.
Dr. Woodruff, I see you have a question. It's just time for a quick question. We'll have more discussion later.
No, okay, I can wait.
Okay, great, thank you. So maybe keep that question in mind till the discussion. So now I'd like to turn to our second speaker, Dr. Dan Wagner, who is assistant professor of obstetrics and gynecology and reproductive sciences in the School of Medicine at the University of California, San Francisco. His research applies the techniques of cell lineage tracing and single-cell transcriptomics to zebrafish embryos as a model for understanding the cellular changes that occur as development progresses. And the title of his talk today is High Throughput Single-Cell Genomics, used to identify genes involved in regulating cellular differentiation during zebrafish development. And I'll now turn the floor over to Dr. Wagner. Thank you.
Okay, thank you so much for the introduction. Can everybody hear me and
Yes.
see my slides well?
Yes, to both.
Okay. So very happy to be here today, learning a lot. So I'm a developmental biologist by training. So I'm from UCSF. I started my lab about 3 years ago. And we study the zebrafish as a means to understand basic molecular mechanisms of cell fate specification in vertebrate embryos. And here on this title slide, I'm showing you two images of a zebrafish. The left is obviously a micrograph showing individual cells labeled by an H2B localized fluorescent protein transgene. The right side shows a data modality that we use extensively in my lab, and that's single cell profiling. So I think Bruce Draper introduced single cell profiling very nicely in his talk. Well, generally what we're doing in these kinds of measurements is we're collecting individual cells, measuring all the genes that are on or off in those cells, and then representing a very high dimensional data set in a two-dimensional plane. So in these graphs, each dot is a cell, and the proximity of the cells on the graph denotes gene expression similarity. And so I'll tell you a little bit about how we're trying to use this mode of data analysis to understand embryonic development in a more quantitative manner than had been possible previously. The main motivations for the research in our group are to understand developmental defects, fate specification, but also how embryos respond to challenges and what sort of processes we might be able to dial up or down to assist embryos in overcoming such challenges. So zebrafish, just like humans, face numerous challenges that can stand in the way of normal development. These challenges span from both genetic causes to environmental causes. And we and others have observed that embryos can have remarkable plasticity. So they can often recover from some pretty catastrophic insults at the genetic or environmental levels. And so we'd like to understand the mechanisms that help modulate the responses to those challenges. As I alluded to on the first slide, so we use a lot of single cell genomics in our lab to interrogate developmental processes. And the reason we use single cell resolved assays are because really the enormous complexity of developmental systems. So every individual cell in a developing embryo has differences. Those differences can be genetic, they can be epigenetic, they could be gene expression, stress, position in the embryo. So there's just a high dimensional vector of differences at the individual cell level. And previously, for gene expression profiling, we've been limited to sort of bulk assays where we would take many cells, analyze the DNA and the RNA in those cells collectively to arrive at a population average. Now, the downside of such assays like this are that we often arrive at non-physiological understanding of the processes because we're averaging between cells and losing important differences. So single cell profiling, by contrast, preserves the individual states of each cell when we make the measurements. And this allows us to gain different levels of insights into the processes happening inside the embryo. So the motivation, as I said before, we want to understand and manipulate mechanisms of cell fate feedback control in embryonic development. Our vision is to combine in vivo studies with tools from single cell genomics. So we use zebrafish in our lab, of course. And I think Bruce and the other speakers today gave fantastic introductions to the system, so I won't belabor this too much. I think Stephanie actually showed this exact same video, which was collected by a former lab mate of mine in our postdoc. But as we've seen, zebrafish are transparent. They develop outside the mother. This makes them extremely accessible experimentally for perturbation as well as for analysis. So I'll just leave it at that because we've well established why zebrafish can be a powerful system. So the methods that we use in my lab to generate hypotheses regarding developmental plasticity and fate specification rely on these technologies for single cell profiling. And these technologies are still fairly recent, probably in the last 5 years is when they took off. They generally use microfluidics, microfluidic droplets or channels, to encapsulate individual cells and make measurements at the single cell level. And then we have an accompanying set of computational tools that we use to analyze these very large data sets and produce graphs or landscapes that give us the ability to understand patterns in these data sets. So I'll tell you a little bit about the technology we use. It's called indrops. This is one of the classic original methods used to encapsulate individual cells into microfluidic droplets. And this allows us to perform barcoded cDNA reverse transcription reactions. Basically, we can create a library of DNA molecules that represent the expression states of each individual cell, and each cell gets a unique barcode that allows us to decipher those patterns when we sequence our data. And so we use this method in my lab extensively through a microfluidic setup, and we can use this to capture thousands of cells from any stage of development that we choose under conditions of perturbation or from wild type embryos. We really have a lot of ability to measure cells in different states when we use the Zebrafish system. So the general sort of computational pipeline that we use for most of our data analysis is schematized here. So as I said, we start with staged embryos. We can grow the embryos to any stage of choice. If we were specifically focused on spinal cord or a brain, we can grow them to a particular stage, collect either the whole embryo or specific cells from the organ of interest. We capture, and using indrops, we record these high dimensional measurements for every single cell in that tissue or in the whole organism. Once we've done our sequencing of these data sets, we've now returned a data set that's very large. It's usually in the form of a large matrix of cells by measurements. And then this matrix can be subject to any number of machine learning data science algorithms. So the way we typically sort of portray these data sets is using a graph like the one I showed on my first slide, or the one Bruce showed, the beautiful depiction of the germline in Zebrafish, where we have little dots. Those dots are individual cells, and those cells can be thought of as points in a high dimensional gene expression space. So this graph here I'm showing has 3 dimensions, but in reality, we have tens of thousands of dimensions. And we generally use the machine learning algorithms to simplify this high dimensional data set into something that we can see with our eyes. So a few years ago, we collected a large data set that described the normal development of the Zebrafish embryo. So this was published in 2018. And in this data set, we analyzed a few different strains of embryos for the first 24 hours. So this takes us from cleavage to gastrulation to epiboly in the beginning of organogenesis in Zebrafish. And so as you can see, we assembled the data into one of these graphs. In this particular graph, I'm showing each cell is labeled by its time point of origin. And I hope you can appreciate that at the early time points, the picture is relatively simple. All the cells of the 4 HPF, that's 4 hours after fertilization. The cells are generally not that different from each other. They form this sort of this ball at the middle of this graph in dark blue. But as we move forward in time, the cells become more different from each other. They become more molecularly diverse. And the algorithms that we use to assemble this graph actually were able to recapture these trajectories. So these branches that extend forward in time. And these branches actually correspond to different lineages of the embryo as they're developing from this sort of early pluripotent state. And so this type of depiction, this depiction of developmental biology, this sort of landscape view, is really the basis of all the work in my laboratory. And so I'll give you some very, very high level quick vignettes about how we use this landscape representation for different kinds of questions in Zebrafish development. So I don't have time to go through full stories for each of these, but I'm just giving a flavor of the kind of quantitation that we can now bring to Zebrafish and to our understanding of developmental biology using data sets like this. So I'll tell you just a little bit more first off about how we mapped all the cell states of this landscape. and what we learned just from the shape of that landscape, the shape of the manifold that came out. And then in number 2, I'll tell you a little bit about how we incorporate lineage measurements, a separate method we can use to incorporate genuine lineage measurements into these cell state measurements. And this allows us to gain some insights about the paths that cells take as they move forward in time across this landscape. Probably of most relevance to this meeting is something I'm very excited about, and that's how we are using this landscape as a powerful phenotyping tool. So most of the data I'll show you today is from wild type embryos, but we can also collect single cell profiling data from perturbed embryos and ask any number of different levels of questions about how a perturbation, and that can be a drug, it can be a mutant, has affected the developmental ontogeny of the embryo. And then finally, I'll tell you a little bit about some unpublished work that we're excited about, where we're interested in gaining some molecular insights into how time is controlled in embryos. So what dictates how fast embryo cells move across this landscape during the process of embryogenesis. Okay, so just starting again, just big picture, what can we learn from these landscape representations of an embryo? So we have detailed molecular data now for each cell from many time points all across the embryo. Because we have this transcriptome information, we can annotate our landscape with any kind of marker gene, so a gene that we might know something about. So here's an easy one. Nanog marks the pluripotent cells. These are the cells of that four-hour embryo that I talked about before. So pluripotent cells right at the center of the graph. So we used markers of different lineages to annotate all the different branches on this landscape, on this graph that we found. So we had markers of the neural plate, the eye, epidermal lineages, mesodermal and endodermal lineages, blood vessels, neural crest. And of interest to this group, we did find germ cells. Germ cells were probably one of the least interesting lineages we saw in this time window of development. So we found germ cells. They didn't change too much as far as we could measure in the first 24 hours, but they were there and they were easy to collect. Interestingly, we're not only, and this goes to the sort of high dimensional nature of these data sets, we're not just collecting sort of identity information about which cells belong to which tissue. This landscape view, this high dimensional view of development captures other information as well. So one of the things that we get for free is positional information. So in the 24-hour embryo, which is the final time point in the data set here, we still have expression of genes that are positional markers in the embryo. So CDX4 marks the posterior of the body, and this is true for many different lineages, so mesoderm, ectoderm. And we can see that our machine learning graph that we've generated actually encodes this information as well as the cell type information. So the posterior cells that express high levels of CDX4 are organized on this landscape in a particular manner. And reciprocally, we have markers for anterior tissues that are expressed in a complementary fashion. So we've done a lot of annotating of this data set. We've made public portals. In the last few years, many groups have actually published similar data sets, so data sets at different times, a few perturbation data sets. And we really have just now a wealth of data in the public domain that gives us detailed molecular information about how all of these different cell types, what defines them, and also what are the changes, what are the transcriptional changes that coincide with their formation during development. And so one of the cool observations we made from just looking at this data, this is wild type data, was related to the topology of cell fate specification. So typically in our textbooks, ontogeny or lineage is represented as a tree. So this bifurcating tree of progressive cell fate choices that lead to very diverse cell fates as you go forward in time. And what we found is that that's actually not the case in terms of the transcriptional identity of the cells. There are many branching regions of the tree, but there are also looping regions. So there are cases where cells that differentiate, but then might have different origins, but then start to look more similarly. So this was particularly the case for the cranial neural crest, which we know differentiates into mesenchyme in the head region and the pharyngeal arches. But I think what our data showed that was a little surprising to me personally, at least, was just how similar those transcriptional states were, to the point where they become indistinguishable. So some interesting insights about topology kind of came from just analysis of the shape of that landscape. So what is it good for, and what are we using it for? The zebrafish embryonic landscape we found does not resemble a tree. So we don't use the term tree to describe this data representation. We either use manifold or landscape. So we can collect data from many different embryos, and we generally get reproducible information, despite the fact that it's an indeterminate lineage. I can go more into that if anyone's interested. And we have sort of in our analysis pipeline, we have no presumption of topology. And this really opens us up to ask questions about converging differentiation, de-differentiation, transdifferentiation, things like this. So those are things that we're very interested in my lab. Okay, so one of the initial reasons we did this is that we wanted to build trajectories that describe the detailed molecular instructions for making any tissue in the vertebrate body plan. And we thought in the early days, oh, we'll just sort of look for these branches, we'll build what they call trajectories, and we'll read out gene expression information for every single tissue and organ and reconstruct the molecular history. And it turned out this was not so easy to do just from the single cell RNA sequencing data alone. And in some cases, it was okay. So the two examples I show here are the epidermal lineage as well as the retina, the eye. And so there are a few cases on our landscape where we could try to draw a path, and it made sense that it represented sort of a stepwise process by which cells are differentiating along a particular path. But there were many places in the landscape where the data were messy or just confusing, and it was very hard to establish directionality. So which way would cells be moving across this landscape? So there are emerging toolkits for building this type of information back into RNA-seq data sets. One of them is a computational method called RNA velocity, which can be very useful. What we used to tackle this was genuine lineage tracing measurements. And I'll tell you a little bit about how we do that and the kinds of things that we're hoping to do next with lineage tracing information. I'm actually going to skip this slide just due to time. The lineage tracing method that we've incorporated into our single cell profiling is called TracerSeq. It's based on a transposase system, the tool2 system, which we routinely use in zebrafish to build transgenics. So what tool2, the transposase enzyme, allows us to do is insert a cassette of DNA into the zebrafish genome, and we can control exactly what goes in that cassette. So in the case of TracerSeq, the cassette is a transgene. So it contains a promoter and a reporter for a fluorescent protein. In this case, it was GFP. And then in the 3' UTR of that transgene, we've placed a barcode, a unique barcode. So we're actually not injecting one species of DNA, we're injecting a library of DNAs into the embryo. So we co-inject this library into the single cell stage with our transposase mRNA. And what we've shown is that TracerSeq works very well by inducing unique insertion events into the genome in this asynchronous fashion. So in this diagram here, I've shown just a small portion of the lineage tree of the embryo. And what we've depicted are integration events. So each of these colored boxes is a particular TracerSeq barcode that's been inserted into the genome. And now anytime that cell divides, the descendants of that cell will inherit that same barcode. And we have many barcodes that get integrated into many different cells, and sometimes into descendants of a cell that was previously barcoded with a different insertion. And so this produces very rich data sets that we can actually combine with our single cell RNA-seq landscape And this allows us to now have ground truth information about which cells are most related to which other cells and where those cells ended up in their differentiation trajectories. So these data sets are very big. We use them in lots of different ways. One of the things we can do with these data sets is analyze individual clones and how cells that are different, how cells differentiating within a clone, within a clade, diverge on the landscape and become different. We can also aggregate data from many different embryos and many different tracer-seq integrations to come up with aggregated pictures of which lineages are often more related to each other. And this has produced very satisfying trees. Now, I know I told you that development is not always a tree, so we have a few violations in this tree. But in general, we've been able to use this data to corroborate places where the single-cell RNA-seq data both conforms to a tree as well as when it violates that sort of tree assumption. So now we have just these two interdependent pieces of information that allow us to establish ontogeny. The tracer-seq lineage data has also allowed us to resolve some complex scenarios. So that looping event that I told you about before, where neural crests appear to be transdifferentiating into a mesodermal-like mesenchymal state. Our tracer-seq data confirms what we know from the developmental biology textbooks about which direction cells might be moving through this complex topology. So this particular loop, our tracer-seq data tells us that there's particular mesenchymal cells that have a neural plate origin that have transdifferentiated from crests into something that looks very much like mesoderm. So these are just a few examples. I don't really have time to tell you about some of the things we're doing with tracer-seq now, but just very briefly, we're combining lineage barcodes with perturbation barcodes, and we're also using tracer-seq to examine how lineage relationships change under conditions of perturbation. But even in the first pass, which I just went over now, the tracer-seq lineage data has been very helpful in helping us resolve ontogeny on the landscape. It's confirmed tree violations, and it's also given us quantitative ways to sort of look at lineage similarity and lineage trajectory. Okay, I'm going to talk a little bit about the next way that we've used these landscapes, and that's to assess perturbations. And really our long-term goal is to understand not only how embryos become defective in response to genetic or environmental cues, but how they could recover from such insults and what molecular mechanisms they might use to recover. So just to show what we can do with this kind of data, I'll describe a really simple experiment we did, which analyzed a classic patterning gene, that's cordin. Cordin is a BMP inhibitor gene that affects the early dorsal ventral patterning of the early vertebrate embryo. This is all vertebrates, zebrafish included. And so what we did is in cordin, because it's a BMP inhibitor, when you inhibit cordin, it leads to overactivity of BMPs and a ventralization phenotype. And so what we did with cordin is we did our in-drop single-cell RNA sequencing. We collected individual cells from control embryos as well as embryos where we'd knocked out the cordin gene using CRISPR. And we compared the transcriptomes of the cells that we got from each of those two samples, and we had replicates back to our reference landscape of wild-type embryos. And what we found was encouragingly what we expected. So we found that in the cordin mutants, ventral tissues were expanded at the expense of dorsal tissues. In this plot, what I'm showing you is I've broken down the embryo into distinct clusters of cells, and these clusters correspond to particular tissues. And I've colored the tissues based on where they sit in the embryo. So whether they're generally in a ventral location or a dorsal location. And what you can see is that on the y-axis and the x-axis of this particular graph, I'm showing you how a particular tissue changed from going from a wild type normal control scenario to the cordon defective scenario. And I hope you can appreciate that the ventral tissues have all moved to the right on the x-axis. And what this means is that those tissues were expanded. There were more cells in those tissues than there were in the control. And the opposite was true for the dorsal tissues. We can also show this the same data by projecting these cells back onto that landscape. So this landscape representation that I've been showing throughout the talk. Now, instead of coloring cells by time point or by gene expression, I'm coloring cells based on how abundant a local neighborhood was in the cordon mutant embryo relative to the control embryo. And so here, purple regions of the graph correspond to regions that were overrepresented in the cordon mutant, and green regions correspond to regions that were underrepresented. And so what we see is that this dorsal ventral transformation is actually, again, it's organized on the landscape. It's not just sort of random, but it corresponds to particular branch points, which we think are cordon sensitive fate decisions. And so this was a really cool result that sort of confirmed what we already knew about this pathway, but it also suggested a relationship to fate choice that wasn't so obvious. And we also found examples of cordon-dependent cell fate switches that occur later than the classic early embryo DV axis specification. The other result that we got from this was that although the predominant change that we saw with the cordon embryos was this changing of cell type ratios, so how abundant or not abundant a particular tissue was, the individual cells themselves actually look almost essentially identical from a normal embryo. So this graph on the bottom left is a measurement of how similar those gene expression profiles were at the single cell level between either our control or cordon and our reference landscape. And you can see similar distributions for these. So this led us to a conclusion that, at least in this particular perturbation, we've affected a patterning change in the embryo. We've skewed ratios along particular fate choices, but because each individual state looks essentially normal, we haven't actually created new states or new pathological states by modifying cordon. We've simply changed how cells flow through these cell fate decisions, and they're flowing differently in a predictable fashion when we knock out cordon or overexpress BMP. So this has led to a number of questions. We have a couple projects in the lab that are sort of examining in further detail what's going on with cordon. But in the final couple minutes, I want to talk about a new project that we've started more recently, and that examines the landscape from one sort of different perspective, and that's through time. And so I've sort of shown you the landscape with time points and how, as cells advance over time, they're becoming more differentiated, more diverse at the molecular level. But we're interested in understanding what really controls the speed at which cells are making these decisions. And the speed or tempo of zebrafish development is something that we've long known can be altered by the environment. So Chuck Kimmel back in the 90s and some of the early staging observations that he made noted something that we all use, probably every lab, is that the speed of development is very sensitive to temperature. So zebrafish are not warm-blooded. They depend on in the environment for their temperature. And we know, we've seen it many times, that at higher temperatures, development proceeds more quickly than at lower temperatures. So there's sort of environmental influence that changes the rate at which cells move through the developmental process. And it's actually quite fascinating that this can work. I mean, we're not talking about one cell type, we're talking about dozens or hundreds of cell types that coordinately know how to remain synchronized at faster or slower speeds at different temperatures. And so it's not only temperature. So we've been investigating this. We've seen a few other environmental conditions that can modulate tempo. One of them is oxygen. Another is simply crowding the embryos, which we think could be oxygen, but there's some evidence that it might be something else as well. And so we embarked recently in the lab on a chemical screen to see if we could get some molecular insights into processes that might be affecting tempo, sort of tempo regulation. So we performed a chemical screen using this Cayman Chemicals that had this particular library we purchased off the shelf. So it had 160 drugs, so a fairly modest screen that was targeted at metabolic enzymes. And so glycolysis, citric acid cycle, and I can talk about why we targeted this, but this was just sort of a first attempt to see if we could modulate this process using drugs. So we used a fairly simple metric to determine whether tempo had been affected. And this goes back to Chuck Kimmel again, but the measurement of the head trunk angle. So as an embryo, as a zebrafish embryo in between 18 hours and about 30 hours is finishing segmentation and organogenesis. The head straightens basically. So as this angle between the head and the trunk increases, it's a fairly linear readout of where that embryo is in time. And so we use both head trunk angle as well as embryo length as a quick readout for developmental progression. And we exposed zebrafish to this chemical library at 2 concentrations between the hours of 19 and 25 post-fertilization. And we did this because this is the window when head trunk angle and length have very linear relationships to time. Let's see, and we, just because this came up earlier, we did this on decoordinated embryos. So here's just an example of sort of the head trunk angle measurement and how this fairly simple thing that we can measure using a micrograph corresponds linearly to time and as well as developmental progression. And so we performed the screen in a few different ways at a few different doses. And what we were looking for in the end were drugs that caused a tempo deceleration, both according to head trunk angle as well as length, and ideally at multiple concentrations of the drug. And so we found a few hits. We're just beginning to investigate the sort of molecular context of these different hits, but we've implicated a few processes that I think are pretty interesting. So there's electron transport chain, there's catecholamine methylation, NAD, NADH metabolism, and one hit that was really related to autophagy. And so we're excited about where this is taking us. I'll say that when it comes to tempo, we're also doing single cell RNA sequencing right now to try to understand what happens in embryos that are going at different speeds. So we're approaching this question from a few different angles. So I think I'm like a couple minutes over, so I'm going to wrap up now. Hopefully, I've been able to give you an impression of how we can use our quantitative single cell approach to understand the process of development in a sort of new way, in a quantitative way that lets us make specific statistical measurements and to do so across the embryo, both in normal conditions and in ways that allow us to infer ontogeny using lineage barcodes. We can use this approach to understand the details of perturbations, and hopefully we'll get some insights into timing mechanisms as well. And so I just want to quickly thank the group, my lab. The tempo project that I talked about at the end is carried out by a grad student, Chris Chen here, and his summer student, Sarah Faust. And so just want to thank the lab and our collaborators at UCSF, who we work with on a number of projects. And thanks to all of you. So I guess I'll answer any clarifying questions that may have come up. Thank you.
Thank you, Dr. Wagner, for that fascinating talk. Just looking to see whether we have any raised hands for clarifying questions for Dr. Wagner.
Okay.
Patrick.
Okay.
Yeah, thank you so much for this wonderful talk, fantastic talk. I had a question. with regarding the loss of function work that you did, so with Noggin, right? So you can capture by single cell these pretty dramatic changes from a functional perspective. But I was wondering what that looks like when you do perhaps when you look at hypomorphic situations where things may not be as dramatic, or when you do pharmacological interventions, if you've tried that, and whether the single cell data still gives you clear answers, or whether it gets a little bit messy because cell annotation is tough, and doing single cell in general is very computationally challenging.
So I'll say that the results we've gotten from single cell analysis of perturbation data have been a bit surprising. I would say that I'm not aware of a case where we have seen nothing yet, but I think Corden demonstrated a principle that that I think has been borne out in many other studies by other groups too. And that's a first observation I think is just interesting, and that's that perturbations that exert massive patterning defects on the embryo's body plan, so dorsalization, ventralization, loss of entire organs, or states that are completely lethal, that look terrible when you actually look at the embryo under a microscope, that the individual cells in those embryos often still look quite normal. And so that's not to say that the proportions are normal, but the actual granular cell states themselves look generally normal. So this is something we're really interested in exploring more systematically. So some of the projects in the lab were comparing sort of different loss of function phenotypes to gain of function phenotypes. So I think gain of function phenotypes or overexpression phenotypes are probably more likely to produce novel states or pathogenic states. And we're very interested in how an embryo would resolve states like that. So I think in some ways we didn't really see what we kind of expected to see, more aberrant states, and we just haven't seen that. And that's been true for other groups as well, generally for loss of function mutations. So we're gearing up right now to do single cell analysis on some drug perturbations, starting with retinoic acid, and I guess we have yet to see if that's going to be a similar case here. But yeah, I think the one thing that's been very, very robustly seen in single cell analysis is sort of whether specification fails or not. So that's something that's easy to see.
Thank you.
I will say one more thing related to that too, and that's that these single cell measurements are just a particular set of measurements we could do. This is the transcriptome. So there's a lot of other variables related to cell identity. So there's epigenetic signatures, there's post-translational modifications. We're not seeing any of those things. So to the extent that changes in a particular pathological context eventually feedback and change the transcriptomes, we'll see that. But there are things that we wouldn't expect to be able to see with this.
Thank you. I see we have a question from Dr. Baskin.
Hi, Larry Baskin, fantastic talk. More of a technical question. You showed some beautiful data that early on in embryogenesis, the cells were basically the same, and then you move out, 4 or 5 days, and very, very, very different. How do you know where you're getting the cells from? And are you using markers, for example, for mesoderm, ectoderm, endoderm, or is it just where you place the needles, so to speak?
So in general, we can do surgical enrichments, and we've done that for a few experiments. For the data I showed today, we're actually dissociating the entire embryo. So we're taking all the cells from everywhere, and we're allowing our computational algorithms to put it all back together based on transcriptional signatures. And so then we use markers in silico to sort of determine which clusters of cells go where. And so I just showed a few little examples of some marker genes that we used to say, okay, this branch is expressing all the mesoderm markers we expected, and this other branch is expressing all the epidermis markers. And generally, it's when you get into those data, it's very convincing because the cells or the cluster doesn't just express one marker; it expresses dozens of markers that are all specific to a particular branch. So yeah, we've generally done pretty well allowing the data to tell us which cells are which. I don't know if that answers the question, but
it kind of does. It seems like it's a little more conducive to the earlier embryos as opposed to later on when you're have a lot of blood vessels and all types of different. The complexity, I guess, increases, but in the system of the zebrafish earlier on, it seems like this is pretty kind of spot on.
Yeah, and I think the the loop that I talked about, where cells that have distinct ontogenies, as they differentiate, they start to look more like each other, even though they're not related in their lineage. This is bound to increase dramatically after 24 hours. So Hox genes are only expressed for so long. So some of the markers that allow us to distinguish these spatial domains, we don't expect those to remain on. And so we may see a collapsing of these things. But within this particular time window, you're right, it is pretty good. I think if we wanted to preserve other identities or spatial origins, we can turn to other methods or include other modalities in this. So optogenetics to mark cells of a particular region such that they would have a label that we could read out with our transcriptional profiling and sort of see that in the data as well. Surgical techniques, there's sort of ways to go about it if needed.
Thank you.
Okay, thanks. Dr. Pessa.
Hi, a very nice presentation. I was wondering, have you segregated on sex, but you don't know the sex at that point, and different lines of zebrafish, do they converge? Does the machine learning algorithms converge on a common pattern?
So let's see, the answer to the first question is we don't know the gender. We haven't looked for that at all yet. It's too early. The second question is, yes, we actually had two strains in our initial data set, and I didn't show, but they lay on top of each other almost perfectly. And another data set that hasn't been published yet by a colleague, they've seen the same thing. So I think, that's something interesting I didn't really go into, but we often think about or wonder about strain differences, but at least at this level of transcriptional similarity across tissues, the signatures that distinguish tissues from each other, at that level, we haven't really seen any trends strain by strain.
Great. Thank you. So it looks like we don't have any additional clarifying questions, so then we can move into our discussion part 2 with both Drs. Panlilio and Wagner. And we have about maybe about 30 minutes for this. So to get us started, maybe something to think about, some suggested questions for discussion. So in the absence of mammalian and mechanistic data on effects of chemicals like domoic acid or bisphenol A, could zebrafish screening studies for general and/or neurological effects alone have indicated the need for additional studies to fully characterize toxicity and mechanisms of action? So that's one thing to think about. Another question is, to design appropriate zebrafish studies, what other types of evidence could help define appropriate lines of experimentation? So for example, with the domoic acid and bisphenol A, we have documented adverse effects on wildlife that correspond to those observed in humans and test animals, and which can then suggest detailed laboratory examination of adverse effects and mechanisms. And finally, another thing to think about is, should zebrafish data generated for the purpose of acute fish toxicity testing conducted for environmental hazard assessment be more generally incorporated into consideration of likely adverse harm to humans. So those are some sort of food for thought, and any discussion from members of the committee or from any of our speakers, including from this morning, want to kind of riff on any of those, or have some other points of discussion they'd like to bring up. I know Dr. Woodruff had a question earlier today that she deferred. Would you like to start with that?
You're calling on me because everyone is like, whoa, that was a lot of really amazing content. Okay. I'm listening to your questions and I'm thinking through. Yeah, I guess I had a couple of thoughts, right? So, I mean, you guys presented a lot of really detailed and, biology and mechanistic information, and kind of trying to think how that, I'm thinking through that kind of piece of it, and some of the, I mean, it's interesting to look at the different ways to measure influences on development. You both talked a little bit about this, including the response or how fast the head unfurls, and but I think when we're trying to think about how to use this information, it all points towards, and what I'm hearing from you is that these are different ways to interrogate development using the zebrafish model. That, and maybe in the domoic acid, I think it's, I wanted to follow up on this question, is that, wait, that's definitely a toxic chemical, and that a lot of the work that you were doing was looking at these windows of susceptibility. And I thought what was pretty interesting about that, which was follow up on the question, Ulrike, that I had, which was you were looking at acute exposures, but if you had done a chronic exposure across all the windows, you still would have identified that effect, right? Because what you saw is that, I think it was post-fertilization 1 and post-fertilization 4, you get kind of a mild effect that was higher at the high doses, but at the 2, everything was depressed. So I think, when we're thinking about trying to interpret data for the kinds of chemicals that we're looking at, it's useful to see you can identify windows of susceptibility, but a chronic exposure could also, right, is what I would take if you had exposed your embryos chronically, you probably still would have seen it, but you wouldn't know exactly when what was the timing that was influential. Right? Is that right?
Right. And I think so, in addition to that, so that's right. So like, let's say that would have required me essentially to be micro-injecting every day from 1 to 4. But then one thing that may do is that may miss sort of lower dose exposures that can happen in a single hit. That's my first thought on that. But most importantly, what I like about using windows of susceptibilities, it also gives you a clue for the mechanisms of action, right?
Right.
Because if you're exposing them through the entire period of time, you don't really know which neurodevelopmental processes are potentially targeted by that. So part of the reason why I did that was like, okay, so we know that the developing nervous system is a target for domoic acid, but what within the developing nervous system and what processes are perturbed? And so that sort of gives you a little bit better of a clue as to like what we need to be watching out for, like kind of going to that guide question about how we can use zebrafish to understand like, the mechanisms by which we like a toxin or toxicant works, right? But then
because I'm kind of, sorry, go ahead.
No,
okay, well, I'm just, well, I guess what I'm thinking is, and it's both those examples, is that from the purposes of where we're looking at environmental chemical exposures, that you could, if you don't hit a particular mechanism that you have been, as you are working through these experiments, if you don't hit that particular mechanism and you don't evaluate that, you could miss a chemical, as opposed to, if you're doing this chronic exposure for domoic acid, and you have an experimental condition that I think is a little bit broader, so you make sure you don't miss something. It's kind of I think our, when people have been asking questions about exposure, and that was interesting about how you did your dosing. That's very important. And the chart, I think we, as scientists, are super interested in all the shape of the dose response curve and the mechanisms. But our charge on the committee is that it can be a response at any exposure. So even though you have these higher exposures, I guess you weren't really sure, or there hasn't been some experiments done to look the difference between the injection versus if you stuck them in a dish of the.
Right. Actually, no, there has been. So I will say, so two things. So it also matters what type of injection. So the paper that I referenced above, where they do, it's much more common to do yolk injections, for example, at earlier stages. To see any sort of effects, they had 3 to 260 fold higher doses they had to use. So I think method is important. And also I did try waterborne exposures, and we did up to 40 micromolar of domoic acid when we didn't see any effect whatsoever. So I do think that, the problem is, of course, this is not high throughput at all. And that's hard. So it would be, for example, I suspect missed in a screening process to do this work because I was not able to see it in waterborne. And this was through a chronic exposure from, I believe I started at 1 DPF all the way to 5 with my highest dose of 40 micromolar. And so, yeah, so that is, we talk about exposure and exposure route and how that's important. So that's part of it for sure.
Yeah, but you said also that they saw this in the animal studies, right? Of response.
Which part?
For the domoic acid. Did you mention that? Oh, sorry.
Yeah, absolutely. So they did. So so for example, in rodents, either they injected the mother, right? And they looked at the, or they injected, neonates, much younger as well. And so I will say, what was kind of assuring, right, is that we saw at least on the very acute neurotoxicity side, we saw very similar phenotypes. So that sort of allowed us to think more about using fish as a model to look at this.
Thank you.
All right, thank you.
I wanted to comment on this thread a little bit too, just about the sort of potential benefits or use of the methods I talked about today, and sort of where they fit into these kinds of goals. So I think, Stephanie Padilla talked about this a little bit, and it sort of depends on the goal of the experiment, how many animals you use, what level of resolution we choose to ask a mechanistic question. So, because our single cell methods, they're getting cheaper every day, but they're still fairly expensive. It's never your first experiment. So we do the same dose response curves, even for CRISPR or mutants. We establish using morphology the general phenomenology of the defect first. And then when it's very reproducible, we use our method to go in and get deep mechanistic insights into what's specifically going wrong under a very small number of conditions. We can't effectively apply this technology to 1,000 conditions. We can apply it to a few. I would say one area in which the single cell approach could be very powerful and maybe different is that it can be good in assessing defects that might be mysterious or pleiotropic, or where it's just difficult to nail down with markers like one at a time, like which sort of tissues might be affected, or if multiple tissues are simultaneously affected by a perturbation. Those are things that we can actually evaluate pretty easily, and without even the need for a hypothesis. So, we just look at every single gene in every single cell and then ask the fish, what changed. And so I guess we could be more open-ended in terms of mechanism, as long as we know we're looking at the right time and place. So I don't know if that helps, but that's sort of how I see a method like the single cell fitting in.
I was kind of thinking also, but your your approach could identify kind of unique pattern signals that could interrogate later, right? Rather than doing more broader evaluation, you could perhaps target some of your exposures also on particular unique patterns.
Absolutely, yeah.
Thank you.
Thanks. Irva, you've had your hand up for a while. I think you might still be muted.
Yeah, I wasn't muted anymore, but I
Okay.
was trying to get the video on. It seems to have a lag. Okay, so I guess this has been really interesting because I have to say that I tend to be skeptical as we get more and more away from humans and then away from mammals and then away from, whole organisms where when we talk about exposures, which is, what DARTIC is about, where, the models seem, to be getting further and further away from what we're trying to do. But I will say that I have been really impressed, and I think the bottom line that I've kind of come out with from today and all of the presentations, really, beginning with Bruce's comparisons of the organs, and all the way through Stephanie's presentation and Jennifer, yours, and Dan's as well, that there are a lot of similarities that make the comparisons I think useful, and certainly from the perspective of just hazard identification, which is sort of that first step that we are, as a panel, are supposed to be trying to take based on the best available evidence. And I understand that there's been sort of the level for in vitro testing has been is always seen as being it can be confirmatory, but and so forth. And I still think that the translation from species to species needs work to establish when things translate and when they don't. And when people bring out that, I don't know, one of the examples earlier on, I can't remember what the chemical was, but you got negative effects in one set of experiments or one species and then positive in the other. And that tends to erode the confidence that we can use that data, that's going to actually be useful for our purposes. But I think the kind of careful mechanistic step-by-step sort of laying out of biological responses and the consequences of those responses and the pathways that follow one after the other, these consequences, is the kind of data that, to me, does speak to the relevance for regulatory purposes such as the Prop 65 that we are working under. So I think that's really, it's not really a question, but just a comment on that. I feel I've really gained a much better understanding that the zebrafish is more than just, there's this transparency and you can see things happening internally, from this. And that's I mean, I think that's kind of the question before us is what is the utility of the zebrafish model? And I see that it's really got a lot of features that make it very relevant, and that the right kinds of studies can really help us in the regulatory realm.
Yeah, thanks. And I think that gets to at kind of one of the questions that we were that I read through at the beginning, which is this idea that using a model like the zebrafish model could potentially be used as screening, in screening for various different kinds of effects, and that might indicate the need for additional studies to characterize, more mechanistic studies to further characterize the mechanism of action of a toxicant. Dr. Pessa.
My question is for Dr. Panlilio. I was wondering, so you found a critical window at 2 days post-fertilization for domoic acid. Would there be any advantage to letting those fish go past day 10 when they've got a more complex array of functions that you can test? Is that something in the work? Because I think that would be very informative.
Absolutely. It was actually a slide I took out, but thinking about sort of the long-term consequences of setting up a nervous system and perturbing it during these critical windows and looking at what that looks like in adulthood is absolutely critical. The one thing that is a little bit harder is that when we start going to adulthood and just with complex behaviors, we don't know as much about the sort of underlying neural circuits for a lot of these behaviors. And I will argue that while there are some really wonderful labs and wonderfully established behavioral protocols to test for example, adult behavior or adult endpoints, it's a little less established than for example, mouse behavior in some ways. And so while we can do that and we can test, for example, adult behavior, what does this mean in terms of, okay, yes, we know that there was a perturbation prior in development, and now let's say there's this deficit in social behavior that we see in adulthood. How do we connect that prior insult to that later adult behavioral endpoint is still tricky, just because we know less about the circuits that drive adulthood. I mean, just because that's, there are still blank boxes there, that doesn't mean it's not important. And I do think growing the fish up and doing that sort of study is important. And I do believe there are people who are doing that.
Okay, thank you. I know we are supposed to take a break of about 15 minutes. And I'm wondering whether this would be a good time to do that. So if I'm not hearing anything from the staff that we should not take a break now, why don't we go ahead and take our 15-minute break. And then when we come back, we'll be talking further, discussing more about the use of zebrafish data in developmental and reproductive toxicity health hazard assessment. So it is 3:02, so why don't we come back around 3:15 and then reconvene then. All right. Everyone have a good break. Okay, welcome back, everyone. We're now moving on to part 3 of our discussion and session today, and that's on the use of zebrafish data in DART health hazard assessment. We have about an hour, up to an hour, to discuss this among ourselves and with all 4 of our invited speakers. So we have several questions that we could discuss and think about. So starting with the first one, given that biological differences between zebrafish and mammals, for example, lack of internal fertilization and pregnancy, what are the issues that should be discussed in considering zebrafish data in human health hazard assessment? Another is, how might considerations of life stage and windows of susceptibility be used in evaluating data from zebrafish? What are some ways routes of exposure and toxicokinetics could be considered in interpreting the results from zebrafish assays? So those are some things, some points that we might want to discuss. Also, applications of the zebrafish model focus on upstream essential processes in reproduction and development rather than final apical outcomes, and how should this be considered when we consider different data streams? And finally, another question to think about is, given the diversity of zebrafish study types and outcomes measured, how might these studies be best evaluated for quality of the studies for purposes of hazard and risk assessment? So if any one of our speakers or panel members have thoughts about any of those, please go ahead and raise your hands, and I will call on people. Let's see, I believe that's, yes, Patrick, Dr. Allard.
Yeah, thank you. I guess what I'm struggling with when I'm thinking about mammalian development and zebrafish development is to think about the timing of things, and thinking, for example, of that's just one example, the blood-brain barrier formation, and whether it forms in a way that's analogous to the generation, the birth of the same neuronal types that we're going to then try to examine and compare with in mammals. So I guess this question is for the panel members, and maybe we can just think about the blood-brain barrier formation. What do we know about it, and how is the timing comparable or dissimilar between the two, and what are some of the related to that? What are some of the limits to the extrapolation that we can make between zebrafish and mammalian species? Should I direct the question to one of the speakers?
Yes.
I think this would be for Dr. Panlilio, perhaps.
Hi, it's a really interesting question. I've thought about this. So, I mean, it occurs slowly over time, where the largest chemicals start getting excluded at 2 dpf. So I was thinking about that in terms of my windows of susceptibility. And so, but it is a really interesting question to think about. Okay, so how does that relate? So I know how that looks like in terms of how does the blood-brain barrier formation occur in fish relative to the developmental processes in fish? One thing that I think this question is really interesting, because I'm trying to think about now how that may be uncoupled, for example, with myelination that happens, in humans, that occurs like well into, early adulthood, right? And so you can imagine that there are potentially processes where it will, depending on the structure of the chemical, that, you'll see it in fish and you won't necessarily have to worry about it as much in humans. It's not necessarily true for domoic acid, just it's a very small, neurotoxin. But it is definitely something to consider, is like, again, not only exposure route, but like also like, how does a chemical, toxin and toxicant, like, how is it able to get to target tissue? And, and one thing about domoic acid too, in particular, is there are specific brain regions that we know of in adults that are more, that are are more targeted. So for example, the hippocampus, because of how domoic acid can sort of shunt that blood-brain barrier. So, I mean, again, so even if it does form over time, and that's an important consideration, there are also still target tissues that sort of allow for more accessibility even without that. So I do think it's an interesting question. I don't have like an exact graphed out answer for how, you know, different processes neurodevelopmentally parallelize with that, but.
Thank you.
Dr. Pessa.
I guess I need to question. I think I heard that in the injection model with domoic acid, the dose may be high, but it's a single exposure, and we don't really know the pharmacokinetics, but you do get a very, very robust response. And I believe the response based on the data I've seen and read about. But then you also just told us that if you put domoic acid on postnatal day 2 up to 40 micromolar in the water, that you don't see any effect. And this goes to my comments last year when we were reviewing a particular set of compounds where I said, well, these studies seem to lack face validity. And then the question came up, well, what is face validity? It means that what you think you're doing at the very fundamental stage, which is the exposure stage, really is occurring. And we heard about, well, small molecule domoic acid has a favorable log P. It should be getting in past the chorion if we understand that the chorion has pores, and yet it doesn't do anything, at least in this context. When we then review papers, how are we to know which ones have face validity and which ones don't? Because unless you have internal concentrations and target engagement as part of that data set, it may not be relevant, I guess. And that goes for a lot of animal studies. I mean, not just the zebrafish. I like zebrafish, by the way. They're great.
Any, Dr. Padilla.
So Isaac, I've thought a lot about this because I started working with zebrafish at about the same time I started screening chemicals. And screening, and I thought, oh, zebrafish is going to be such a big change, and it is. But screening chemicals is a really big change too. And you have to get comfortable with not knowing everything. That's the first thing. And what I've always said with chemicals and screening is there's lots of reasons to get a negative. There are just lots of reasons to get a negative, and you really have no idea why you've got the negative. The positive, on the other hand, you do have something happening. And so usually I view the positives with a lot more certainty than I view the negatives, because there's just, I mean, the chemical could have fallen apart. The chemical could be sticking to the plastic. The chemical could be sticking to the chorion. The chemical could be sticking to the outside of the embryo and never get inside the embryo. It could be breaking down. It could be pumped out by reverse pumps in the zebra. I mean, there's all kinds of things that could be happening to give you a negative. But the positive means that something has happened, and let's go after it, or let's let somebody else go after it. But I guess that's one of the things. And if you're only looking at negatives, then you either need to know the chemical is there in the fish, or else you need to have, the other thing you need to have is some positive controls. So that's another thing that you need to look in papers is are they running positive controls? Can they pick up a chemical if it's a positive? And some papers don't do that.
I agree.
I mean, this is all pretty obvious, but for me, it was a different mindset when we started doing screening.
Thank you. Dr. Wagner, did you have a comment on that?
Hi, sorry, I had a comment on the previous topic. I just wanted to mention the blood-brain barrier before we got too far away from it. So, yeah, there has been some really cool recent work done on the zebrafish blood-brain barrier by Dr. Natasha Brown and Sean Megason. So basically, the barrier forms at 3 dpf, and there have been some elegant studies that show what you can do. Tracer dextran injections into the bloodstream to investigate when the barrier forms and which regions of the brain and spinal cord become protected and when. And I believe some of the molecular regulation that's been uncoupled so far supports a lot of similarities between the zebrafish mammalian systems. So it's a combination of transcytosis and tight junction regulation that dictates what size molecules can get through and when. So there's a literature on that. I'm not personally an expert in it, but it is out there. So I think you could definitely approach questions of barrier penetrance in the zebrafish if you had the right timing and knew where to look.
My understanding is it's controlled by many of the same molecular mechanisms too.
Yeah.
Yeah, thank you for that. Dr. Woodruff.
I just want to make sure I got that. I understood that you said that the blood-brain barrier formed 3 days post-fertilization. Is that what you said? That wasn't my question, but then when you said that, I wanted to make sure I understood that.
I think it's 3 to 5 days, but I'd have to check to be perfect.
But it's after fertilization, some day, number of days.
Yeah.
Okay, thank you. I just want to follow up on Stephanie's point, and just going back to, the ability of the zebrafish or exposures in the zebrafish to be predictive of what we might see in other species. And I thought your presentation on chlorpyrifos and BPA were pretty illustrative. Those are 2 chemicals that we've already declared as a committee as developmental reproductive toxicants, and they seem to be well conserved in your model. I wonder if you could comment on that, or in that I feel that I agree about the exposures, but I also think that thinking about chronic exposures is really important for us, because that's typically what we are looking at in the population of humans, is not one-time exposures. That can occur with some of these chemicals, but for many of the chemicals that we're evaluating, it's a chronic exposure. And I think that also the information you presented on concordance in general with other zebrafish models is also very useful for us, because I think that I think zebrafish is an underutilized tool for us for evaluating toxicity of hazards. And I think just thinking back all the way to the beginning presentation about conservation of development across species is generally true, or majority or super majority true, I think helps us, I think zebrafish should be, placed more in our wheelhouse for evidence that we're evaluating for toxicity and hazard.
So I actually don't really remember collecting the BPA data. All I know is it was positive.
Oh, you showed some on the
I know,
yeah.
But as I said, we collect most of our data in a blinded fashion. For the chlorpyrifos is a chemical that I studied in many different ways, and I had a lot of it. So, and it tends to be very stable in DMSO. And so we decided to use it as our positive control. And it's very interesting because we were checking these plates every day. And I always would get sucked into thinking that the chlorpyrifos positive control wasn't working, because the fish would look completely normal up until about day 5, which is about the time that the liver comes on board. And so as soon as the liver comes on board and the chlorpyrifos gets converted to the chlorpyrifos oxone, then the fish begins to go downhill developmentally. And by day 6, we always see either death or malformation in the chlorpyrifos animals. And we've also run other people's chlorpyrifos. We've also run chlorpyrifos oxone. And so it is extremely toxic, very early in development. And so it appears to be behaving. We also use it, we haven't talked about this, but I also use it as a positive control for my behavioral studies in the zebrafish. So even when we dose at levels that don't cause any developmental effects in the zebrafish, we still see behavioral effects. And the behavioral effects, if we keep them until they're 14 days old, are even more pronounced at 14 days than they were at 6 days. So it seems to be a chemical that is behaving as you would predict it from the human and mammalian laboratory animal tests in the zebrafish. But I mean, that's one example.
Thank you. Any other thoughts or questions on that topic or other topics? We can think about it while I ask whether we have any public comments. Did we have any comments from members of the public or questions? See.
And if a member of the public would like to make a comment, please use the Zoom function to raise your hand.
We haven't received any in the chat, I don't think, or I guess they can, or through email.
We do have one hand raised.
Let's see. I'm not, let's see. I'm not seeing
So,
that.
Okay, well, I can allow that. Oh, well, it looks like they just lowered their hand.
Oh. All right. And we don't have any to read either, is that right?
Okay, they've raised their hand again, so
Sean,
I'll let that person speak.
Great.
You have 5 minutes.
Okay, thank you for all these presentations. They were really wonderful. My name is Sean Burgess. I am at the University of California, Davis, and I am also a colleague of Dr. Draper, who spoke this morning. I just had one comment about the chronic exposure versus the kind of more immediate exposure that was just mentioned. Yeah, I think that one point is that, you know, during development or during human development, there will always be some stage where they're being exposed to whatever they're being exposed to, and it can have an effect. So I think that, you know, thinking about chronic exposure is important, but I also think you can think about how you can have kind of acute exposure just because, you know, we develop and experience all stages of, you know, embryogenesis. And any one of those could be sensitive to one of those chemicals. So I was wondering if anyone could comment on that.
Thank you for that comment. Did any of I think you mentioned one of the speakers, do any of the speakers want to comment on that? I mean, certainly, I don't think we should discount acute exposures. You know, obviously, you can have different information based on the exposure paradigms, and that obviously is something to think about. What are the exposure paradigms that maybe would be most useful in this type of an application? And I think there was an argument made for chronic exposures, but certainly, as we saw from some of our speakers, acute exposures can also have very pronounced effects. And I see that Dr. Padilla has her hand raised.
So, I mean, one of the things that we've often thought about, sort of like what Sean is talking about, is one day in the life of a zebrafish embryo is a very long time when you think of it compared to a human embryo. Or, I mean, so it's hard to quantify those differences and to equate them. So, I mean, so much happens in a very, very short time for zebrafish development that you know, in an entire human trimester could be a day and a half. And so it's going to be hard to equate those and decide how you feel about them.
Thank you. Any other thoughts from other panel members or speakers? Dr. Allard?
Yeah, I guess I'm going to react to some of the comments that I heard earlier, and forgive me if my comments are not necessarily well put together, but I heard the comment moving away from the point of interest and you know, thinking about the concordance sometimes between rodent species, between a mouse and a rat, or you know, thinking about the lagomorphs as well, like bringing in the rabbit, and the concordance between each one of them and humans. Those concordances are sometimes not great either. And using an evolutionary approach where we look at multiple species at the same time to really identify the critical chemicals that seem to alter the same developmental pathways or similar developmental pathways across phyla is actually, to me, a much more powerful tool to really identify something that will be potentially potent in humans as well. Yeah, the other thing I think that's to me that's also important to think about is that I think we're all kind of making, or some of us, including me, I think we're making comments about you know, replicability between laboratories and whether that's concerning or not. But this problem of replicability, at least I'm sort of heartened to see that you know, several labs look at the data, analyze the data together using same tools, which is often lacking in vertebrate data, which also suffers from replicability. The lack of repetition. I cannot say it, but you understand what I'm trying to say. And we've seen that on this panel before, right? We look at this data and we have X number of studies showing a positive outcome and X number of studies showing a negative outcome, and then we use weight of evidence. It's kind of part of the process of weighing those things. But at least with zebrafish data, what I'm again heartened to see, and I'll repeat myself, is the fact that then you can take large data streams where people have screened large amounts of chemicals, large number of chemicals, and then use similar methodologies to analyze this data and compare it. And I think this is actually very powerful.
Thank you, Dr. Allard. See, I just saw that someone else had their hand raised, but I think they lowered their hand. Any other comments from other committee members or from any of the panel members? Dr. Baskin.
You know, looking at all the zebrafish data and then getting, you know, incredible lectures today, it seems like it's going to be a very important model. And I was kind of just asking staff of OAHA, would the future possibly be that this would be kind of our first line screening tool, and then if there was a question, we would still go to, you know, mouse rodent models, or do we think that this is going to replace the use of mouse and rats?
So are you asking for the staff to respond to that?
Or whoever would like to comment on it, because you know, I just, the zebrafish is still a zebrafish.
Dr. Sandy, did you have a comment?
Yes, for OEHA and others can jump in, but I'd just say that we're not proposing that we would use this as a screen. or a first step, but we're just acknowledging that there are more studies out there in the literature when we do a search for DART effects on with a particular chemical. We're running into, we've been presenting it over the last few years, data on zebrafish. And we anticipate that we'll see more and more data in zebrafish. So we thought it was useful to delve into this topic in more detail today.
Thank you. Dr. Woodruff.
Yeah, I would hope that we see zebrafish presented as another stream of evidence along with our human and mammalian evidence. I mean, the data presented today, I guess I would not say just screening, but it's a line of evidence along with what the other pieces of evidence that we have, and the presenters, including using it to interrogate developmental biology that's relevant to humans. So it feels that this is an important source of information for us to be evaluating as we're deliberating on our decisions.
Yeah, thank you. And of course, we're also evaluating mechanistic data streams too,
Right.
Culture models, and putting it all together.
And there's mechanistic data in the zebrafish data.
In the
A lot
zebrafish
of
data, yes.
mechanisms.
Yeah. Dr. Padilla.
So I'm sort of sorry to talk so much, but I'm really enthusiastic about this. And so one of the things that we haven't talked about today is there is a whole literature on using zebrafish to discover the underpinnings for human disease. And there's been very successful. There's a whole literature about tank to bedside, where there's very little laboratory mammals in between. And that's also very interesting and may give you more faith in some of the decisions that you want to make. And then also I wanted to respond to Dr. Baskin's remark. As somebody who's been trying to convince the regulatory community of the usefulness of zebrafish, one of the things, and you sort of alluded to this, the first thing is sort of a screening and prioritization, right? We have 100 chemicals. Which ones are the worst? Which ones are the worst actors? And so, can we identify those and then test them in our more familiar laboratory animals? And then as the Europeans are beginning to move to, actually regulating on human toxicity using zebrafish data. But for that, of course, you need to understand exposure. So these are very common problems that many, many other regulatory agencies are trying to struggle with. And in my experience with the EPA, 10, 15 years ago, they really weren't too much interested in zebrafish data. But now, because there is so much out there, as you all were talking about, you do a search and all of a sudden you've got all these zebrafish papers. There's so much out there that it kind of behooves people to figure out how it is useful, which is, I know what you all are struggling with, but everybody else is struggling with it too. But there is a whole literature out there using zebrafish to determine mechanisms and mechanisms of disease and identifying chemicals that could possibly treat disease that we haven't even touched on.
Thank you. Dr. Hertz-Viciotto.
Right. Yeah, I think several people have already, hit on some of the main issues, and just somewhat reiterating and maybe putting a little different light on my earlier comments. I find what Stephanie just said to be really interesting. That, I mean, obviously we're focused on developmental reproductive. That's this committee's charge. But the use of zebrafish widely now in a lot of diseases. I think, you know, and I should be looking at that too, does increase, you know, some confidence in utilizing these data. And, you know, I continue to be curious, not just about dose, but routes of exposure and that entire, you know, unknown, and is there uncertainty really about how to compare across routes of exposure, which, and in fact, you know, really some of the questions of for some of these exposures, what data do we have about, you know, low level human exposures? You have to kind of look at it chemical by chemical, and there's still, we have a lot on some chemicals and maybe a lot less on some others, including, you know, domoic acid, it seems. But, you know, with all of those caveats, the basic question of does this particular exposure pose a potential, does it have the potential to cause reproductive or developmental harm? It still stands as valid evidence in living systems, including organisms that share quite a bit with the humans, and 70% of our genes. So, and the genes regulate our responses to you know, exogenous insults. So from that perspective, you know, I think, you know, obviously it requires looking at all the mechanisms, looking at exposures, looking at routes, looking at and the endpoints, and how, you know, what do those what was it, the Mothin, I've already forgotten the name of the cells.
Wagner.
Yeah, do the correlates respond similarly to similar types of triggers? And, you know, those are all answerable questions, you know, with maybe some certain kinds of experiments that are a little different, just, you know, slight perturbations of current sorts of experiments, which, I mean, that's the beauty of experimental science, that, you know, as an epidemiologist, I can boast that, oh, it's relevant for the people we're trying to protect directly. But boy, do I get envious when I hear some of these presentations where you can, you know, have a very specific question, you can answer that question, then take that result, and that raises the next question of, okay, then does this, you know, the next step in the pathway. So yeah, it's compelling in many ways, and I really appreciate the presentations and today really opening up that issue for us on this panel.
Thank you. Dr. Allard.
Sorry, I know I talk too much. I guess I would just like to reiterate the point made earlier, is that as the number of studies are growing, again, exponentially, if you look at PubMed, and we're probably going to review a lot more of that data in the future, I think it's very important to have expertise related to non-canonical or alternative animal models, including zebrafish especially, both on the staff side and perhaps on this panel as well, so that we can really easily tell apart a high quality study that used sound methods for people in the field, as opposed to the opposite, not sound methods. So being able to tease those things apart will require expertise. And I'm not sure if we have it right now. It'd be great to know whether we do or not. But if we don't, then we definitely need to have that in-house.
Thank you. I'm just looking to see if there are any additional comments from panel members or from any of the presenters? And certainly we've heard a lot of really great presentations today about the utility of using zebrafish to both to understand mechanisms and to potentially and also for screening for toxicity. And I think we've heard from several panel members too, that given the increase in the number of studies in zebrafish and also just the utility of looking at various different model systems and multiple streams of evidence from mammalian systems and the fish models, as well as potentially other models, and the utility of comparing those systems, and especially if there are similar signals that come out in multiple model systems, how that strengthens the evidence for reproductive and developmental toxicity, since as is our focus. So if there are no other comments and I don't see any additional raised hands, then I think that we can check one more time with Julian whether there are any additional public comments, and if not, I think we can move on to the next item.
I'm not seeing any at this time.
All right, great. Thank you very much. And I'd like to thank all of our speakers again too. And now we're going to move on to the consent item, which is an update of the California Code of Regulations, Title 27, Section 27,000, list of chemicals which have not been adequately tested as required. So we are now ready to take up this consent item. The committee is being asked to affirm changes in response to submissions from the Department of Pesticide Regulation. The US EPA has indicated that there are no changes. This is a ministerial duty of the committee, and that we rely on the information provided to OEHA by the Department of Pesticide Regulation and US EPA in order to identify the chemicals that need to be added or removed from the Section 27,000 list. I'd like to introduce OEHA Special Assistant for Programs and Legislation, Jillian Lichty, to give the staff presentation on this item. Jillian.
Thank you, Dr. Luderer. So Proposition 65 requires the state to publish and update annually a list of chemicals that are required to be tested under federal or state law for carcinogenicity or reproductive toxicity and have not yet been adequately tested as required. This list can be found in Title 27, Section 27,000 of the California Code of Regulations, and is commonly referred to as the Section 27,000 list. It's separate and distinct from the Proposition 65 list of chemicals known to cause cancer or reproductive toxicity. The Section 27,000 list has no regulatory impact. It does not require that any testing be done. Rather, it's a source of information concerning chemicals that need further testing pursuant to state or federal law. To update the list, OEHA requests information from the California Department of Pesticide Regulation and the U.S. Environmental Protection Agency's Office of Pollution Prevention and Toxics and Office of Pesticide Programs each year. This year, OEHA staff reviewed these responses and identified one recommended change to the Section 27,000 list: removal of bromodiolone. Based on information received from DPR, data requirements for this compound have been fulfilled, and further carcinogenicity and reproductive toxicity testing are not required. The letter from DPR, along with additional background, response letters from US EPA, a mock-up of the proposed change, are available in the staff report provided to the committee and posted online on September 30th. The proposed change is also shown on this slide. As Dr. Luderer mentioned, this is a consent item and a ministerial duty of the committee, and that the DARTIC and CIC committees use the information provided by DPR and US EPA to identify the chemicals that need to be added to or removed from the Section 27,000 list. We ask the committee members to vote in favor of the proposed change so we can update the list. Unless you have any questions, I will now turn it back to Dr. Luderer.
Thank you, Julian. So again, are there any questions from any panel members? Okay, not seeing any raised hands. Then I'll read the question. So the question we're voting on is, should section 27,000 of Title 27 of the California Code of Regulations be amended as indicated in the staff report? And I will now call your names and ask you to vote yes, no, or abstain on this question. Dr. Allard.
Yes.
Okay, Dr. Ayun Kim, she may have.
Yes.
Yes, okay. Dr. Baskin,
Yes.
Dr. Carmichael,
Yes.
Dr. Hertz-Piciotto,
Yes.
Dr. Pessa,
Yes.
Dr. Plopper,
Yes.
Dr. Woodruff.
Yes.
Okay, and I also vote yes. So I affirm that we have I guess the required, we have a unanimous yes vote, and I think 6 yes votes are required to affirm the change. So based on that, we have more than 6 votes. All right, and next we will move on to the staff updates. We'll close out this meeting with these updates on the Proposition 65 listings, regulations, and litigation that have taken place since our last meeting. So I will again ask Julian to present on the chemical listings and safe harbor levels.
Thanks again, Dr. Lederer. So we are providing you with an update on important Proposition 65 developments since the last DARTIC committee meeting. I'll start by going over the chemicals or endpoints added to the Proposition 65 list or under consideration for potential listing, as well as data columns requesting information on chemical toxicity. Then I'll discuss adopted and proposed safe harbor levels. After that, I will turn it over to our Chief Counsel, Carolyn Rowan, who will provide an update on other regulatory actions and significant Proposition 65 litigation. Next slide, please. Since the committee's last meeting, 5 chemicals have been added to the Proposition 65 list. PFNA and its salts, trimethyl propane triacrylate technical grade, tetrahydrofuran, methyl acrylate, and 2-ethylhexylacrylate. Additionally, the cancer endpoint has been added for 2 chemicals previously listed for reproductive toxicity, PFAS and its salts and transformation and degradation precursors, and PFOA. Next slide, please. 2 potential cancer listings are under consideration. At the December meeting of the Carcinogen Identification Committee, the committee will consider whether to list bisphenol A as causing cancer. This chemical was first added to the Proposition 65 list by the DARTIC in 2015. Finally, antimony trivalent compounds is under consideration for listing administratively under the Labor Code mechanism. Since the last DARTIC meeting, OEA has issued a data call-in on bisphenol S to request information related to its reproductive toxicity. This information may be used in the preparation of a hazard identification document. The data call-in ended last April. BPS was one of the chemicals prioritized by the DARTIC in 2020. Next slide, please. Since the committee's last meeting, safe harbor levels have been adopted in regulation for a carcinogen. And no significant risk levels were adopted for oral and inhalation exposures to 1,3-dichloropropene 1,3-D and became effective October 1st, 2022. We also proposed a safe harbor level for antimony trioxide and are reviewing comments received on the proposal. And now I will turn things over to Carolyn.
Thanks, Julian. So on the first slide here, we have other Prop 65 regulatory actions from the past year. The first one on the list is safe harbor warning for cannabis smoke and benign THC exposure. These regs provide non-mandatory specific safe harbor exposure warning methods and content for retail products that can expose consumers to cannabis smoke or delta-9 THC via inhalation, ingestion, or dermal application, and for environmental exposures to cannabis smoke and delta-9 THC businesses where smoking of cannabis or vaping or dabbing of delta-9 THC occurs. So this regulation became effective on October 1st, 2022. We also have the safe harbor warning for glyphosate. And this regulation provides safe harbor guidance for businesses that cause exposures to glyphosate from consumer products that require a warning. And this regulation was recently adopted and will become effective on January 1st, 2023. Next up is the safe harbor warning for acrylamide in food. This regulation also provides safe harbor warning content. This one's for businesses that cause exposures to Prop 65 listed chemicals in food and beverages that require warnings. And we submitted it to OAL last month, and we expect to hear back on this proposed reg by October 28th. And finally, we are actively working on exposures to acrylamide in cooked and heat processed foods. On October 6th, we noticed a second modification of proposed text and an addition of documents and information to the rulemaking file. So this regulation is out for comment at the moment. And litigation, I think, is on the next slide. Thanks, Julian. We have 4 cases that are currently active that I can provide updates on today. One is the Physicians Committee for Responsible Medicine, or PCRM, versus Newsom. And that case is a challenge to OEHA's decision not to list processed meats. We're in the discovery stage right now, so we haven't reached any merits briefings or hearings, so that's the update there. We also have the National Association of Wheat Growers versus Bonta case. This one involves a First Amendment challenge to the glyphosate warning requirement. The challenge centers on the argument that because only the IARC has identified that chemical as a carcinogen, and other agencies, including US EPA, have said it is unlikely to be a human carcinogen, there can be no warning that would be misleading. The district court determined that required warnings for glyphosate exposures violated First Amendment limits on compelled speech, and the AG appealed to the Ninth Circuit. And the case was on hold while OEHA was preparing its new glyphosate warning reg. Now that that new regulation has been approved, the case will become active again. The parties are going to file one last brief in the Ninth Circuit, and the court will then decide whether to send the matter back to the lower court or proceed to oral argument. We also have Calcimber versus Bonta. This case involves another First Amendment challenge. Here it's a challenge to the safe harbor warning for acrylamide. And the district court previously granted a preliminary injunction and the Ninth Circuit affirmed. The case is back with the trial court, although there has been very little activity since the new judge was assigned. And finally, there's Council for Education and Research on Toxics versus Starbucks. In this case, CERT challenged the OEHA regulation on coffee as part of a long-running enforcement action. As you may recall, OEHA adopted a reg essentially saying that chemicals formed in coffee from the roasting and brewing don't require a warning under Prop 65. The coffee reg was used as a defense in this case, and the trial court upheld the regulation and entered judgment for the coffee companies. CERT appealed, and the court heard oral arguments in September, just last month, and we are awaiting a decision. So that's the status of litigation at the moment.
All right, thank you very much, Carolyn. Our last item, I'd like to ask, we have Director Lauren Zeiss to summarize the committee actions.
I
It
have a
looks like Dr.
question.
Yes.
I just have a
Irva.
question. I just had a quick, can you go back one slide? I was trying to figure out, did I hear this? Did you say that the safe harbor warnings for cannabis were not required? The others were on this slide? Or did I mishear? what you said?
No, the safe harbor warning for cannabis are final and effective now. They were effective October 1st.
Oh, okay. I just misheard something.
Yeah, no problem.
Okay, thank you. And I'll like to turn over to Dr. Lauren Zeiss, Director of OEHA, to summarize committee actions.
Great, thanks, Ulrika. Okay, so we had a fascinating set of presentations and also a really rich discussion on zebrafish. And I think we heard a lot of enthusiasm for continuing to present this strain of evidence, the committee, this stream of evidence. And, you know, I think we'll reflect on the discussions of the committee and speakers as we prepare the material for the hazard identification documents. And we'll really look forward to future engagement on presenting this evidence and also evidence on other NAMs. So thank you so much. It was a very full day and a lot of rich discussion. So thank the committee and the speakers for that, as well as the staff for all the work to put this session together. And a special call out to Marlissa Campbell. So thank you so much. We also a unanimous decision to remove bromodiolone from the section 2700 list. So that was a unanimous finding by the committee. And so, and the committee also heard the updates. So with that, I think we'll wrap it up, and I'll turn it back over to Ulrike. I just want to thank the committee for a really rich discussion and all the participation. Very helpful to us, and I hope it's going to be helpful to you as you consider these new data, especially the zebrafish and other new data streams. I'd like to thank the audience and also staff from all the work to put together this meeting. It definitely takes a village for this, so thank you so much. Back to you, Ulrika.
Yeah, thank you. Yeah, I'd like to echo Dr. Zeiss's comments and thank everyone, the speakers, the staff, the committee members, and adjourn the meeting. So thank you, everyone. Goodbye.